Hello and welcome to today's TechNights sessions. My name is Rochi and I'm a fifth-year PhD student in Computational Biology Department at Carnegie Mellon University.
Hi, I'm Shahul. I'm a second-year PhD student in the same program as Rochi. And today we're going to be talking about single-cell sequencing, which is an exciting new technology in biology, which helps give us a better understanding of how living systems work.
One essential goal of biology is to understand what makes up our human body and how these different components work together to keep the human body functioning. There are different levels of organizations within the human body. First, there are 11 distinct organ systems in human beings. Each system consists multiple organs that work together to perform one or more functions. For instance, the stomach and intestines belong to the digestive system, which helps to break down and absorb nutrients when we eat food. If we look into the organs closely, we would see that each organ is formed by different tissues, and each tissue is a collection of cells. Cells are the smallest and most basic units of a living thing. There are unicellular organisms where their whole body is just one cell, and there are multicellular organisms, including humans like us, where in these multicellular organisms, there are multiple cells come together to support the body collectively. So, how does the cell fulfill all kinds of important functions? It turns out, just as humans consist of multiple organs, each organ fulfilling a specific function, cells consists of different organelles. The word organelles means tiny organs. The largest organelle in a human cell is the cell nucleus, which is considered to be the control center of the cell. The nucleus sends out instructions to other organelles based on a set of rules which are encoded in DNA. Just like the rulebook in school life, where we have individual regulations of different aspects, such as no cell phones in classroom or only athletic shoes in PE class, DNA also has different parts that correspond to a specific aspect. Each DNA fragment that is responsible for a function is called genes. DNA is a really important material in human lives. It contains the genetic information in the unit of genes that instruct all kinds of functions of cells. For us human beings, we inherited the DNA from our parents, and as a result, virtually all cells in our body would share the same copy of DNA. As mentioned, DNA is the rulebook of the control center for the cells. So does it mean that every cell in our body is the same? On the surface level, it seems like it should, but that is not the case. Human body is actually formed by different types of cells. Cells can be classified into different categories based on multiple features. For example, based on the location of the cells, we can label cells as heart cells or brain cells. Based on their functions, we can classify them as cells that helps us to fight disease or cells that provide structure and support, such as fibroblasts in our skin. So, why does the same copy of DNA result in various types of cells?
Okay, so to understand why, we'll first briefly introduce the central dogma of biology. So the central dogma is just a very fancy name for how the instructions in the rulebook actually get enforced. So during this process, genes in our DNA are first transcribed into messenger RNA molecules. As the name implies, these molecules are responsible for conveying instructions from the cell nucleus to other parts of the cell. Then, in those different parts of the cell, the messenger RNA goes through a process called translation, which produces proteins. Proteins are molecules that come in very different shapes and sizes, and they carry out various functions in the cell. For example, some proteins are used to transfer information between cells. Some are used as antibodies to fight off foreign viruses, and others are used as structural scaffolding to build up muscles in our body. So in this way, information flows unidirectionally from the DNA in our cell nucleus to the functional proteins in other parts of the cell. The figure here might make you think that in the cell, one copy of DNA results in one copy of messenger RNA, and eventually just one copy of a protein. And as a result, all the cells in our body would have the same number of proteins no matter what cell they were. However, in actuality, through transcription and translation, one copy of DNA can become many copies of messenger RNA. And then many copies of messenger RNA can be translated into many, many copies of proteins. So in this way, each cell can have different kinds and quantities of proteins. We call the amount of RNA produced in a cell its gene expression level. And we can measure this expression level for each gene individually in a cell. So back to our question. How can the same DNA result in different types of cells? It turns out that this variability is caused by the different gene expression levels in different cell types. For instance, cell type 1 would have 5 copies of protein A, while another cell type might have fewer copies of that protein. We can summarize the gene expression levels of different cells as a table, and each cell would correspond to a series of numbers in this way. For example, cell type 1 would now correspond to the number 521, and so on. Thus, we can regard the gene expression level of a cell as its ID card. Let's say we have a scanner that can read out the gene expression level of each cell. Then, because different genes have different functions, we can infer what the cell's purpose is just based on the types of genes that it expresses at a high amount. With this magical scanner, we can eventually hope to understand every cell type in our body and the function that those cells have in making our body work. So, what is this magical scanner? How does it work? Well, let's demystify it a little bit. In order to actually identify individual cells, we do a laboratory experiment called single-cell RNA sequencing, or scRNA-seq for short. The first step is to take all of our cells and separate them up so that each one fits in a tiny droplet of water. Once we isolate that droplet, we then cut up the cell, mash it up, and take out everything that's inside. When we cut it up, what comes out is a bunch of RNA molecules, and all we have to do then is count them to get the unique signature of the cell. This all might seem a little bit abstract, so let's try doing it with a real biological system. We're going to do a hands-on activity in which we extract DNA from a strawberry. Note that this isn't exactly the same as scRNA-seq. In that experiment, we're extracting messenger RNA from a single cell, whereas here we're going to be isolating DNA, which is the rule book that we talked about before, from a whole strawberry. However, a lot of the ideas and techniques that we'll use are the same as what you'd use for scRNA-seq. For this experiment, you'll need the following reagents: strawberries, salt, isopropyl alcohol, also known as rubbing alcohol, dish soap, and a cup of water. In terms of equipment, you'll need a strainer, a Ziploc bag, a measuring cup, some spoons, and a couple of glass containers. For this experiment, we need to make sure our isopropyl alcohol is ice cold, so put it in the freezer before you start. Now, count out about 10 strawberries. Note that you don't need gloves, but they are necessary to make you look like a real scientist. Put the strawberries into the Ziploc bag. Make sure to seal the bag tightly so that nothing leaks out. Next, slowly mush the strawberries with your hands by pressing on the Ziploc bag. This may take some time, so keep at it. Make sure to break the strawberries apart as much as possible. If there are any big chunks left, keep squishing. Next, we're going to use one of the glass containers. Pour your cup of water into the container. Then, measure out about a tablespoon of dish soap and mix it vigorously with the water. Finally, measure out a teaspoon of salt and mix it into the soap solution. Now, pour the mixture into the bag with the crushed strawberries. Again, make sure to seal the bag tightly. Now, pulverize the bag with your hands again to make sure that the soap solution mixes well with the strawberries. This may take a few minutes. Once you're done with that, place your strainer over another glass container. Pour your strawberry mixture into the strainer and make sure to remove the big chunks of strawberries that you didn't manage to crush. Next, measure out about a cup of ice-cold isopropyl alcohol. Pour it into the same container. Almost immediately, you should start to see DNA precipitating out of the mixture. Take a utensil, such as a chopstick, and start swirling the mixture. The DNA should stick to your utensil. Now, let it sit for a few minutes. The longer you wait, the more DNA will precipitate. Finally, give it a couple more swirls. Then, prepare a paper towel. Take out your utensil and the DNA, and use the paper towel to dry them out. The sticky, slimy stuff that you get back is DNA. Once you let it dry out a bit, it should look something like this. So there you have it. Now you know how to extract DNA from a strawberry.
Even though we have the single-cell sequencing technologies, using it to identify a cell's identity can still be a hard problem. First, due to technical difficulties and randomness of the gene expression process, the information we collect through the magical scanner can still be noisy and incomplete. Second, in the human genome, there are more than 20,000 genes, meaning that this barcode on the ID card can have more than 20,000 digits. This is called the high dimensionality problem, which makes the analysis for single-cell sequencing data really difficult. Moreover, there are various versions of the single-cell sequencing technologies, or even for the same single-cell sequencing technologies, environmental conditions such as the temperature can affect the information we collected from the cells. Thus, datasets generated from different labs, although all contain necessary information to identify the cell types, might not have direct match. Just as an analogy, for most of your ID cards, they all contain the information such as your name and your birth date, but they might have different format or location on these ID cards. This is called the badge effects. To address these challenges, computational methods have been developed to analyze the single-cell sequencing data. Here, we provide a Google Colab link such that you can go through the process with us.
So we've linked the Google Colab notebook in the description of this video. And in the next part of the video, we're going to walk you through the notebook step by step. If you aren't familiar with Google Colab, it's basically just a way for us to share some code and plots that we created in a way that you can interact with them. You can use the notebook by clicking on the link in the video description and opening it up in your browser. Note that in order to run the code in the notebook, you'll need to be logged in with your own personal Google account. However, if for some reason you don't have a Google account, you can still view the plots in the notebook without actually running the code that generated them. So in this Google Colab session, we're going to introduce you to a typical pipeline for for identifying cell types from single-cell RNA-seq data. We will be using a dataset consisting of measurements from mouse brain cells. Before moving on to analyzing the data, we need to install some software. To do this, just click on the play button on the left side of the code block, and that will install and load all the required packages for you. We'll need to wait a few minutes for the installation to finish. Once it's done, there should be a small green check mark that appears next to the play button. This means that the packages were installed successfully. The next code block is necessary just to make sure that the plots that we generate look nice. So similar to the first code block, just click the play button and wait for it to finish. Next, we have to download the dataset that we're interested in analyzing. So run the next code block, and this may take a minute or two because the dataset is quite big. Okay, now that we've done all the setup, we're ready to inspect our mouse brain cell dataset. Click the play button to plot the cells that we're analyzing. This particular plot is looking at a slice of mouse brain tissue, but zoomed in at really high resolution. It's kind of like looking at the tissue through a microscope. Here, each black dot represents a single cell and its location in the tissue. However, although we can see all of the cells, we can't tell what type of cell each dot is, just based on its location. As we've discussed, we can use single-cell RNA-seq data as a scanner to identify the cell type of each cell. But how can we do this?
As mentioned, the single-cell sequencing data we have is extremely high-dimensional. So how to analyze the data and identify cell types based on that? One important technique is to project the data from high-dimensional to lower-dimensional space. Just as an analogy, we showed how we can project a 3D model of Mario to 2D space from different angles. Through a certain angle, we manage to reduce the dimensions, but still able to preserve the important information such that you can identify that's a Mario immediately. Following the similar idea, we can project gene expression of single cells from high-dimensional space to lower dimensions while preserve important information to separate cell types. As an example, we first have the count matrix representing the gene expression levels of these 3 genes. We can then represent them as individual dots in the 3-dimensional space. Through computational algorithms, we can find a projection that can transform the data to 2-dimensional space while showing clustering patterns. Here, each cluster of cells can be regarded as a cell type. In the real world, we have much more than 3 genes, but the general idea would be similar.
So back to our code. Now that we understand the idea of a projection, let's use it. The next code block runs an algorithm called UMAP that projects our single-cell RNA-seq mouse brain dataset from the high-dimensional space to a lower-dimensional space. And this will help us greatly with identifying cell types. By the way, you might encounter an error message when executing this code block, but don't panic. If you try running it again a few times, it should eventually work. Next, let's visualize the projection of these cells that we just computed. This projection finally gets us a picture of the cells that is useful for differentiating them. From this plot, we can see that there are naturally some clumps or clusters of cells in the projected UMAP space. As discussed previously, each cluster of cells should correspond roughly to an individual cell type. But how do we know which cell type corresponds to which cluster? Well, thanks to previous laboratory experiments, it turns out that we have some prior knowledge about special genes called marker genes. These genes are expressed almost exclusively by specific cell types. So, if we see that a cell type expresses a marker gene at a high level, then it's very likely that that cell is that corresponding cell type. For instance, we know that the gene PLP1 is a marker gene for oligodendrocytes, one type of cell. So, let's color our UMAP projection by coloring the dots in the plot based on their expression level of PLP1. This creates a gradient which demonstrates the expression of PLP1. Another example, we also know that HPCA is a marker gene for the subiculum cell type. And similarly, PTGDS is a marker gene for endothelial tip cells. So, from the colored plots, we can pretty easily identify which cells correspond to these marker genes. Repeating this process for each cell type marker gene combination, expert biologists can create a complete cell type annotation. You can visualize this complete annotation of all known cell types by running the code block below. Now, in this scatter plot, each dot or cell is colored based on its cell type that is annotated by experts. And that's exactly what we wanted. Remember, however, that each cell came from an actual physical location in the mouse brain tissue. Thus, we can map the expert annotated cell type colors back to their spatial position in the brain tissue. By executing this code block, we can see the spatial patterns of the cell types that we analyzed. For instance, we see long strips of the subiculum cells through the tissue. It is interesting to see that some cells prefer to be located next to cells of the same cell type, and some other types of cells are just scattered throughout the whole tissue. Finally, we can plot the marker genes that we used to identify the cell types in terms of spatial coordinates. As we can see, the spatial patterns of the marker genes correspond closely with the spatial patterns of their matching cell types. So that's it for our notebook. Hopefully this example with the mouse brain dataset gives you an idea of how we can generally learn important information from a single-cell RNA-seq dataset. So in summary, our body is composed of many different types of cells. By identifying each cell and its unique characteristics, we can understand how all our cells work together to help our body function. And furthermore, we can understand how they respond to different conditions, such as disease or environmental stress. And one way to differentiate cells is by their differential gene expression profiles, which we can measure with single-cell RNA-seq, as we learned today.
We hope you had fun learning with us, and see you at the next TechNights sessions. Bye.
Bye.